Hi guys, this is Ms. Romani again. You are going to learn about sexual reproduction and a type of cell division called meiosis. Remember what happened to the banana, right? Asexual reproduction produces babies that are genetically identical and all the drawbacks that come with being genetically identical to each other, or in other words, clones of their parents. So in contrast, sexual reproduction creates what is called genetic variation. So instead of every baby being identical to the parent, to the single parent, we have babies that are a result of two parents combining so that their babies are genetically different from each other. And so what we end up is what is called genetic variation. Like, look at this family of cats. In this picture, there is a mom, there's a dad, and there's three babies. And all of them are genetically unique, as you can tell by their fur coloration and their shape and a whole bunch of other details about their physiology and how they look. And that is the key about genetic variation, so that we don't have a situation like we had with the banana. If there's a disease, if there is some sort of obstacle in the environment, the idea behind genetic variation is that even if not all individuals can survive that obstacle, some of them might be able to. And so therefore, it might not increase the survival of the individual, but it definitely can do a lot for increasing the survival of a species. So sexual reproduction produces genetic variation by combining the genes of two parents, usually in animals, a female parent and a male parent, whereas asexual reproduction involves only one parent and there's no combination of genetic material, so the one parent produces identical clones of themselves. And so the idea with sexual reproduction is that each baby is the result of a unique genetic combination of their parents' genes. So I just said that for most animals, the parents are usually male and female. So what determines then if an organism is male or female? Well, in humans, it is the presence or absence of a Y chromosome. Females have two X chromosomes, and males have an X and a Y chromosome. When we take a look at chromosomes in humans, we will find that out of the 46 chromosomes, 44 of them are what we call autosomes, but two of them are sex chromosomes, the X and the Y. The X chromosomes do not include a lot of information about being female. You can see that it is a large chromosome. The larger the chromosome, the more genes it contains. It's a large chromosome, it contains a lot of genes, but very few of them are about being female. Whereas the Y chromosome is a tiny chromosome, and it pretty much just contains genes about making a baby into a male. There's a couple of genes that have to do with height, but the rest of the genes on the Y chromosomes are all about making a baby into a male baby. Whereas the X chromosome does not necessarily have to include information on how to make a baby into a female baby. Female is technically the default sex. So when a baby is being formed, it will be female unless there's a Y chromosome to tell it otherwise. The chromosomes that do not control or determine the sex of the baby are called autosomes, and they contain information about all kinds of traits, whereas the sex chromosome, the X and the Y chromosome, might contain information about some other traits, but are also the ones that are going to determine whether or not the baby is going to be female or male. Males and females produce sex cells called gametes. That is what we call eggs or sperm. And these are produced via a type of cell division called meiosis, which we will learn about in a minute. So, mothers will produce eggs on their ovaries using meiosis, and fathers will produce sperm in their testes using meiosis. The eggs and the sperm only have half the number of chromosomes as the parents. So, for humans, the number is 23. Each egg has 23 chromosomes, one of each type, and each sperm also has 23 chromosomes. All female eggs or gametes will have an X chromosome because females only have X chromosomes to give. Whereas since males are XY chromosomes, 50% of their sperm will have the X chromosome and 50% of their sperm will carry the Y chromosome. And so that helps determine what the sex of the baby will be, depending on which sperm cell reaches the egg first. If it's one that's carrying a Y chromosome, the baby will be male. If it's one that's carrying the X chromosome, the baby will be female. So during fertilization then, a sperm, in this particular case, one that's carrying the Y chromosome, is going to fertilize an egg, which always carries the X chromosome. And this process is called fertilization. And the first cell that is produced from this fertilization is called a zygote. A zygote is basically just a term to refer to the fertilized egg, the first cell of a human body. And then the zygote would undergo several rounds of cell division called mitosis. Remember, we talked about mitosis in the previous lesson. And mitosis is going to produce a baby that is going to be ready to be born in about 9 months. And eventually, mitosis is going to lead to this baby to grow into a full adult, which then is going to continue the whole process. And so, before we move on to the next slide, I want to just clarify a couple of terms. And that refers to a description of the number of chromosomes that can be found within the cells. And the terms are haploid and diploid. So, haploid cells are basically cells that only have one set of chromosomes, or half the number of chromosomes, which in humans is 23. Haploid cells are produced by meiosis. So, which cells are produced by meiosis? Well, eggs and sperm cells are produced by meiosis. So, these would be haploid cells, and they are usually denoted by the symbol N to signify that they only have one of each type of chromosome, so just N copies of the chromosomes. On the other hand, diploid cells are cells that have two sets of chromosomes, so they're denoted with the symbol 2N, and basically all our body cells have a diploid number of chromosomes, as well as the fertilized egg. So after fertilization, if you have a gamete or a sperm that has N number of chromosomes, and an egg cell that also has N number of chromosomes, both of them produced by meiosis, then the zygote, the fertilized egg, will have 2N. N plus N, that's 2N. So for humans, those numbers are 23 and 46. So if an egg cell has 23 chromosomes and a sperm has 23 chromosomes, together the new baby will have 46 chromosomes, which is the right number of chromosomes for a human. So here we go, the baby will continue to be 2N, and then it will grow into a 2N father, which eventually will make haploid or N sperm, which will then fertilize a haploid egg, and the process and the circle of life will continue. So just take a look at just another picture that kind of shows that. Eggs will have one of each chromosome, so one of chromosome number 1, one of number 2, one of number 3, and so on, and one X. Whereas the sperm will have also one of every 23, one of number 1, one of number 2, one of number 3, etc., and a Y chromosome or an X. And then when fertilization happens, then the homologous chromosomes now can combine, and you will have 2 of each type. So 46 chromosomes. So this is a diploid cell in the testes that eventually undergoes meiosis, and same as in the ovaries, and eventually they undergo meiosis to produce haploid eggs or sperm, which then will undergo fertilization to produce a diploid zygote. So during meiosis, one diploid or parent cell will undergo 2 cell divisions to produce 4 haploid and genetically different gametes or daughter cells. So we always start with a cell that is diploid, that is found in the testes in males and in the ovaries in women. Those diploid cells will undergo 2 cell divisions, meiosis 1 and meiosis 2. And at the end of meiosis 2, that one cell will now have produced 4 separate cells, each of them with half the chromosome number that was in the original cell, and each of them genetically different from the other cells. And so that when those eggs or sperm combine, you can have an unlimited number of genetic variation in the babies that are produced. Let's talk about the stages of meiosis then. Fortunately, the stages of meiosis have similar terms to refer to each stage as the stages of mitosis. So you don't have to memorize an entire new terminology. Unfortunately, meiosis and mitosis, because they have similar steps, they tend to be familiar or similar enough that sometimes it can get confusing. So let's go through the stages of meiosis. We always start with intraphase, so we'll start with intraphase. So an intraphase, the cell is diploid, so it has 2N number of chromosomes. The centrioles have duplicated, and chromatin has duplicated and is uncondensed. This is the same as intraphase in mitosis. You can see the nucleus, you can see the nucleolus. The chromatin has made a copy of itself, but it's not condensed into chromosomes yet, so chromosomes are not individually visible. Because meiosis is divided into two stages, meiosis 1 and meiosis 2, we're going to add a Roman numeral 1 after each step in the first stage of cell division. And so this next stage is called prophase 1. Prophase 1, it starts with one process that did not occur during mitosis, and that is a process called synapsis. During synapsis, the homologous chromosomes find each other and pair up. This did not happen during mitosis. During mitosis, the chromosomes never paired up. But during prophase 1 of meiosis, it's important that the chromosomes find their partners or pair up. So chromosome number 1 from mom finds chromosome number 1 from dad. Chromosome number 8 from mom finds chromosome number 8 from dad, and so on. Then an interesting thing happens between those chromosomes that have paired up, and that's something called crossing over or recombination, which basically means that these chromosomes that have paired up are now going to exchange pieces and therefore shuffle their genetics and create more genetic variation. Let's go back to that analogy that I gave you at the beginning of the lesson on genetics, and that is of the recipe books. So each chromosome is like a recipe book, and you have one copy of recipe book number 1 from mom and one copy of recipe book number 1 from dad. During prophase 1, what would happen is that the recipe books would find each other. So both of recipe books number 1 will get close to each other and pair up. And then what is happening during crossing over would be the equivalent of the recipe books exchanging pages. And so let's say pages 5 to 10 from dad's book will go into mom's book, and pages 5 to 10 from mom's book will go into dad's book. And therefore they will shuffle their genes that way and just add for a little bit more variety in the genes that are possible for the babies. The site at which crossing over happens, the place where the chromosomes sort of touch and then exchange pieces, is called the chiasma. So there are two processes then that increase genetic variation in the gametes. The gametes are the eggs or the sperm cells. I already alluded to the first one, crossing over. So let's just kind of take a look at that again. Let's say you have a chromosome from mom and a chromosome from dad. When crossing over happens, basically they just exchange pieces, just like you see here. Just a way of just shuffling their genes, shuffling for increasing genetic variation. And so now you have more variety. Now the other thing that happens is going to happen in the next stage, and it's called independent assortment. And during independent assortment, what that basically means is that when the chromosomes line up at the middle of the equator of the cell, which they will do in pairs, in order for them eventually splitting up into two separate cells, the order in which they line up is completely random. So that, let's say in a situation like this one, where we have two pairs of chromosomes or 4 chromosomes, one possible way that they could line up would be like this, with the dad's chromosomes on one side and the mom's on the other, which would then give us these possible combinations of chromosomes in the gametes, the eggs or the sperm. But another possible combination could be this one, where chromosome number 1 from mom and chromosome number 2 from dad go to one side, and chromosome number 1 from dad and chromosome number 2 from mom on the other side, which then now gives us another possible combination of those chromosomes when the eggs and sperm are produced. So what this does is just gives us more ways by which the chromosomes can be combined. So now when you have just 4 chromosomes in an organism, you can end up with 4 possible combinations. That's basically 2 to the power of n being the haploid number of chromosomes. So in the previous example, this organism had 4 chromosomes, so the n number was 2. So the possible combinations is 2 to the power of 2, which is 4. I know, it's a little bit of math, and it's supposed to be biology, so don't worry about the math. But I just want to show you something. So the n number for humans is not 2. Humans have 46 chromosomes, which means that our n number is actually 23. So do you know what happens if we have 2 to the power of 23? If you take your calculator, you try it, if you do 2 to the power of 23, you will find that the answer is 8,388,608. So that means that whenever humans make their eggs or their sperm, they are over 8.3 million possible combinations of chromosomes in the eggs or sperm that are produced by humans. This is the reason why no two siblings will ever be identical. Because every single time a father makes sperm, there are over 8 million different genetic combinations of his genes to make those sperm. And every single time a woman makes eggs, the same is true. Over 8.3 different possible combinations of her genes in order to make her eggs. And that's not counting the shuffling that happens with crossing over. So there's an almost infinite possibilities of combinations and genetic variation that can happen when humans make eggs or sperm. So, a couple would have to have probably 9 or 10 million or more children for them to possibly even end up with two that might be identical to each other genetically. It is almost impossible. Okay, so after prophase 1, we enter metaphase. We're going to still follow PMAD, prophase, metaphase, anaphase, telophase. So prophase 1, now we have metaphase 1. Now metaphase 1 is different than metaphase in mitosis because in metaphase and mitosis, the chromosomes lined up in a single line down the middle. And in metaphase 1 of meiosis, the chromosomes are going to line up in pairs. They're going to be lining up in the homologous pairs along the equator, not single file. And then this is the time when independent assortment can happen because how they line up might vary depending on just chance. So, which chromosome ends up at which side is just completely random. Spindle fibers will also still shoot out of the centrioles. They'll attach to the centromeres. A lot of the same events that happen during mitosis are happening here, but the main difference is that instead of lining up down the middle, the chromosomes are lining up in pairs. So next, we get to anaphase. And because the chromosomes line up in pairs, now when we get to anaphase, the chromosomes are basically going to separate from their partners. They're not splitting down the middle. They're just separating from their partners. And so the homologous chromosomes are therefore being pulled apart from each other, and the sister chromatins still remain attached at the centromere. But chromosome number 1 from mom is being separated from chromosome number 1 from dad, and chromosome number 2 from mom is being separated from chromosome number 2 from dad. And the reason that this happens is to make sure that every egg or every sperm ends up with one of each type of chromosome. So even though each egg or sperm ends up with 23 chromosomes, it's not just any random 23. It's one of each type. So it would make no sense for us to end up with 23 chromosomes, but you have 2 of chromosome 1 and 0 of chromosome 5. You need one of every type. So that's why the homologous chromosomes pair up and then separate from each other. Next, we have telophase 1, and this is now when that diploid cell becomes 2 haploid cells. So this is where now the chromosomes are going to be encapsulated again into a nucleus, and the cell's going to split in 2, and each of these new daughter cells are haploid because now they each have only half the number of chromosomes as the original parent cell. And so here's just a summary of what meiosis 1 looks like. You can see crossing over, independent assortment in metaphase 1, anaphase 1, telophase 1, and 2 haploid cells. So let's continue from there. So now we're going to skip interphase and we're going to move on to prophase 2 directly. During prophase 2, now the centrioles double, pair up, and spindle fibers reappear. Meiosis II is actually very similar to mitosis. The nuclear membrane and nucleolus disappear, the chromosomes are visible, they never uncondensed, and these cells are still haploid. Metaphase 2, the chromosomes line up single file along the equator just like they did during mitosis. The only difference is that this is happening with 2 cells, and the cells are haploid. Anaphase 2, now the sister chromatins are pulled apart into opposite ends, and again, the only difference between this and mitosis is that it happens to 2 cells, and the cells are haploid. And then last, telophase 2, this is where cytokinesis also occurs. We end up with 4 haploid cells, and they are genetically different from each other because genetic variation was achieved by both independent assortment and crossing over. So you can see if you look at the chromosomes within each of these cells directly, you will find that they are each genetically unique. And I have an animation to show meiosis II. So I wanted to briefly talk about the difference between meiosis in males versus meiosis in females, because when males make sperm and females make eggs, the process is slightly different from each other. As you know, eggs and sperms look very different. So let's start with meiosis in females, and this is a process called oogenesis. Genesis always means the beginning of, so this is the beginning of the egg. And the key with oogenesis that is different from sperm creation is that notice how we start with one diploid cell, but we end with only one haploid egg. And that is because the egg is the biggest cell in the human body. It is the only cell that is actually visible without a microscope. If you want to know exactly the size of an egg cell, a human egg cell, put a dot with a pen on a piece of paper. That's the size of a human egg cell. So it's actually the only cell in the human body that is visible without a microscope. In order to produce this very big cell that is going to provide essentially all the cytoplasm and organelles for the zygote or the baby, you need to make sure that at each step of cell division, only one of the cells ends up with most of the cytoplasm. And so one large egg cell with all the organelles and cytoplasms is produced. And the other cells that are produced are just sort of tiny little cells called polar bodies, which are not going to be anything. They're just going to disappear. They're not an egg. Only one egg is produced per meiosis. And the other thing that's interesting about egg production in humans is that all baby girls, every single baby girl is born with every egg she will produce in her entire lifetime already in her ovaries, already having started meiosis, but sort of suspended at prophase 1 stage. And then after puberty, every month, one of those eggs are going to complete meiosis and be released as a single egg cell. But the interesting thing is that females are born with every egg that they will ever produce already present, which means that a 20-year-old woman has 20-year-old eggs. A 40-year-old woman has 40-year-old eggs, which is the reason why maternal age is so important for the health of a baby. The older a woman is, unfortunately, the older her eggs are. And so the greater the chance that those eggs might have accumulated some mutations during her lifetime. This is also one of the reasons why women sometimes, if they have money and they have the means to do so, might actually freeze their eggs if they're planning on having babies later on in life, so that if they wait until they're in their 40s to have babies, they might be able to use some of the eggs that they put aside when they were much younger and their eggs were, I don't want to say fresher, but yes. Meiosis in males is a little bit more typical than what we saw before. 4 haploid sperm are produced, but the difference is that instead of having just regular sort of circular gametes, a process happens to the sperm by which they essentially grow a tail called a flagellum, and they end up with very little cytoplasm because they only really need just some mitochondria for energy to make sure that the tail can move in order for the sperm to reach the egg, and then the nucleus. And that's basically all they contribute to the fertilization is their nuclei, their DNA. And also the other difference between egg formation and sperm formation in humans is that sperm formation starts after puberty in males. Male babies are not born with every sperm they'll ever produce in their testes. They're actually, sperm are produced fresh daily, starting sometime around puberty. And there is no menopause for men, so men can still continue to produce sperm throughout their lives. And that's that, guys. That's the end of our lesson, and I will talk to you soon. Bye-bye.